First presenter, his name is Dr. Reza Najat. He is going to give a talk about the neuroprotective effect of erythropoietin. Please welcome him to the stage. The floor is yours now, but before you start, you need to give 5 minutes about yourself, because I have not seen your profile or your CV. Yeah, just 5 minutes briefly. And you have 30 minutes for your presentation and 10 minutes for questions. We are a little bit behind the time, but we hope we can move on time. Okay, please welcome him.
Good morning. It's an honor to be with you. And Mr. Chairman, thank you. I'm Dr. Reza Najat, Fellowship of Critical Care Medicine in Iran. I work in Bazargan Hospital, charity hospital. I was a former faculty member in Shahid University of Medical Sciences. Now, because we are behind of time and the time is short, I should start as soon as possible. And the problem of neurological injury in brain injuries is very important. There are many patients suffer from this problem all around the world, and we should help them. You know, I start from the first slide here. Okay, erythropoietin is a growth hormone. It helps erythropoiesis, means to produce new red blood cells. But it is already a growth hormone. And 100 years should have passed that make the scene ready for this growth hormone to be used in everyday practice. Well, EPO or erythropoietin, according to these slides, you will see EPO. EPO, the abbreviation of erythropoietin, is required for survival, proliferation, differentiation of red blood cells, and it prevents apoptosis. Apoptosis means cell death. Cell death. And EPO-R is produced in the kidney in adults and in the liver in the fetus before birth. Both liver and the kidney produce EPO in some of the cells which have the characteristics of neural cells in the kidney and in the liver. So EPO has something within our central nervous system. EPO can be found in many systems in our body: nervous system, cardiovascular, digestive, endocrine, female and male reproductive system, respiratory system, spleen. What does it do? How it is secreted? It secretes according to an oxygen sensed based manner. Whenever you have hypoxia, you have erythropoietin level be increased in your body. And the factor that helps to increase the secretion of erythropoietin is HIF, hypoxia-inducible factor. HIF in hypoxia increases in our body. It goes to the nucleus of the cells. It stimulates hypoxia-regulated element in our genome, and this hypoxia-regulated element or hypoxia-reducible element in our body, in the nucleus, stimulates or activates genes to produce EPO and more than 100 other proteins in our body. So hypoxia makes some proteins available to our body. Erythropoietin, for exert its effects, should have receptors. its receptor reside on the cell membrane, and it is a kind of cytokine type 1 receptor. Erythropoietin itself increases its receptor in our cells. Whenever erythropoietin is secreted and activated in our body, it increases its receptor too. For this reason, it can exert its effect and exert its activity as well as possible. You see, in mice, in animal studies, it could be found that EPO and EPO-R, whenever the animal lack EPO and EPO-R, receptor and erythropoietin and its receptor, apoptosis of cell death occurs before anemia, because whenever erythropoietin is low, anemia occurs. But before anemia occurs, apoptosis occurs. For this reason, it has a cell surviving, cell protecting activity in our body. This is the general aspect of its receptor. This is called cytokine JAK-STAT signaling receptor or pathway system. It is a general thing that occurs in our body. Cytokine bind to its receptor, and then JAK is a transcriptional factor stimulated, then STAT signaling stimulated. STAT means signaling, transducer, and activator of transcription. It means that this protein goes to the nucleus, STAT, and activate or make the cell produce some other proteins. They are anti-apoptotic. It means that whenever erythropoietin binds to its receptor, apoptosis decreases and self-surviving occurs. Okay, EPO and EPO-R, JAK2, mitogen activating protein kinase, and then STAT5, previous slide, if you can remember. It increases some proteins with anti-apoptotic activity. Sorry. And once again, autophosphorylation of this JAK2 after binding EPO and its receptor inhibits pro-apoptotic molecule, and it inhibits, prevents mitochondria to start apoptosis or cell death. Mitochondria is the energy plant of the cell. Our cells produce energy for other cells or itself, themselves, within mitochondria. And once again, apoptosis decreases when JAK2 is stimulated with erythropoietin. Erythropoietin, when it binds to its receptor, also decreases inflammation. I thank Dr. Kelly, who presented us with something very important in our brain, and the patients with schizophrenia. You know, the patient with schizophrenia have some sort of neuroinflammation, and apoprotein was found to decrease inflammation within our brain. Okay, another activity or another property of erythropoietin, it increases ENOS. ENOS means endothelial. Endothelium is the cell which reside within our vessels. And NO is nitric oxide. S means synthase. It means that erythropoietin increases NO in our vessels. NO is needed for autoregulation, or better to say, increase the blood flow within our vessels. So whenever you have a patient has ischemia within his brain, erythropoietin might increase the blood flow to the ischemic part, and it may have the activity it may have the activity for the brain to increase its blood flow. Okay, after lots of articles published for the cell surviving and cell protecting manner or activity or property of erythropoietin, a multi-center study rejected all of them and told us that administration of erythropoietin had no cell protective effect and even might be hazardous. But this is the article published in 2009. After that, lots of articles published exactly in the opposite direction of this multi-center study. Like this, that erythropoietin could abolish hypoxic-induced central respiratory depression, 2015. And in 2016, it is said that erythropoietin could diminish MRI brain injury in neonates. And in mice, it was found that when EPO-R is knocked out, neural progenitor cells are cells which reside in our CNS, central nervous system, that can move to the injured side and produce natural, or better to say functional neural cells. It is a regenerating or neurogenesis, but they work in neurogenesis. So, without erythropoietin receptor, no low neurogenesis occurs. You know, erythropoietin receptor is not homogeneously spread in our central nervous system. Somewhere has more erythropoietin and somewhere has less. In limbic system and hippocampus, there are lots of erythropoietin receptor. Erythropoietin is produced de novo within the tissue or the central nervous system. And it has paracrine or autocrine. Paracrine means a cell produces erythropoietin, the neighbor cell uses it. And autocrine means a cell produces erythropoietin and it exerts its effect over the cell, the producing cell. So we have many areas in our central nervous or brain which produce erythropoietin and use it in autocrine or paracrine fashion. Their expression of EPO and EPO-R is low in healthy people. But when injury into the central nervous system occurs, it increases. Yes, blood-brain barrier is a barrier that prevents ions, toxins, and other molecules to reach to the parenchymal cells in our brain. Erythropoietin is a big molecule. Does it pass through BBB or blood-brain barrier? Yes, when injury occurs, blood-brain barrier disrupts, so erythropoietin can reach to the parenchymal cells. Another thing, that EPO and EPO-R are produced within our nervous system too. It means that it is not needed erythropoietin, systemic erythropoietin reach to the parenchymal cell of the brain. Whenever oxygen supply to the brain is low, HIF, hypoxia-inducible factor, increases and increases EPO production within the brain. But without hypoxia, mechanical damage, infection, metabolic stresses, low glucose or insulin, oxidative stresses, elevated temperature, intense neural activity, and enriched environment may increase erythropoietin production within the brain. And pro-inflammatory cytokines. Cytokines are molecules with messages to our brain. Pro-inflammatory, they produce inflammation within the brain or within other parenchymal or systems. But in the brain, any pro-inflammatory molecules which produce inflammation within the brain decreases erythropoietin. So, CNS lesions, when it occurs, it has a primary, exactly the necrotic, the most affected part with necrosis, and it has secondary injury. We have an area, you know, we have a core infarcted, cell death occurred. We have an area in the circumference called penumbra. In penumbra, cells reside, although they are alive, they cannot do their job in healthy manner. In healthy manner. We should help penumbra to live, to survive, in order to decrease the lesion, the injury. Okay, we have secondary injury. Secondary injury means that propagation of the injury to the penumbra. When you have lesion, when you have injury, like ischemic hypoxic or even hemorrhagic, ischemic hypoxic, like a stroke, you have a primary injury, which is the stroke. You have secondary injury, which is inflammation. Inflammation, according to the previous slides, decreases EPO. So you have a penumbra with a live cell, but they cannot do their job. Inflammatory cytokines decreases erythropoietin there. If erythropoietin, we can prove that has a cell-protecting effect, we should give the penumbra erythropoietin to have the cell-protective effect. Okay. And whenever an ischemic part of the brain, in the ischemic part of the brain, blood flow is regenerated. It is called reperfusion. And within the reperfusion time, oxygen stress occurs, free radical occurs, excitotoxicity, and nitric oxide production, according to the previous slides, nitric oxide increases in reperfusion, but within the reperfusion, nitric oxide is hazardous to the brain because it increases the inflammation. And blood-brain barrier also is injured within the stresses that occurs within the brain. We have inflammatory mediators, reactive oxygen, and some hormones called vascular endothelial growth factor. The growth factor which increases the endothelial cells within the brain, within the injured site, and matrix metalloproteinase. You know, when the injury occurs, the body would like to increase the vascular production. But this vascular production, or the canals which should go through this injury, should have an environment to have the possibility to increase. This environment is full of debris.